Good afternoon, everyone. Welcome. I'm Debbie Hart, the President and CEO of BioNJ, and I'd very much like to thank you for joining us today for an exclusive look at BioNJ's newly published white paper, Health Equity in Clinical Trials, New Jersey Participant Demographics, developed in collaboration with Medidata, a Dassault Systems brand and the industry leader in clinical trial solutions. The release of Health Equity in Clinical Trials, New Jersey Participant Demographics, is a key milestone in BioNJ's Health Equity in Clinical Trials initiative, launched in 2022 to ensure health equity for all. As we know, clinical trials are the source of every new therapy and represent a $15.2 billion industry in the US alone. Yet, for all the promise they represent, substantial inequities in clinical trial enrollment limit the benefits to society at large. This research addresses a vital goal: improving recruitment efforts in New Jersey's clinical trials by evaluating demographic representation and comparing recruitment metrics to similar states, including Connecticut, Maryland, Massachusetts, and New York. We're excited to have our experts here from Medidata to share key findings, discuss recruitment trends across states, and highlight essential public policy steps needed to achieve demographic representation that mirrors epidemiological data. Today's webinar is being recorded and will be available tomorrow at bionj.org. We encourage you to ask questions by placing them in the Q&A box. And now, to lead the discussion, it is my great pleasure to introduce BioNJ's Vice President of Government Affairs, who also led this project for us, Dr. Ian McLaughlin. Ian, over to you.
Sure. Thanks very much, Debbie. And thank you all for joining us today. As Debbie mentioned, this project focused on clinical trials and specifically on quantifying demographic representation across participants in trials that were conducted in New Jersey. Again, my name is Ian McLaughlin. I'm the VP of Government Affairs at BioNJ. Before joining BioNJ, I worked in health policy in the state's legislature. And then before that, I actually did my PhD at the University of Pennsylvania in neuroscience, where I developed a deep appreciation for the disparities in health outcomes that communities across the country confront, as well as underrepresentation across the various nodes in biomedical science and research. So I was thrilled to get to jump into this project, particularly because it's intended to address one very significant dimension of those disparities. And so, the project is really intended to get a sense of trial recruitment performance among communities in New Jersey. And as Debbie mentioned, those participant data in New Jersey were compared to data that characterize recruitment in states that are the most statistically comparable to New Jersey, which again, as Debbie mentioned, are Connecticut, Maryland, Massachusetts, and New York. State similarity was based on everything from demographic composition, of course, but also things like infrastructure and geography and a bunch of other criteria, because while the challenges of recruitment in a state like New Jersey are going to share some characteristics with any other state, it'll definitely present unique opportunities and challenges when compared to a state like Wyoming. And just to put a fine point on it, New Jersey is the densest and one of the most diverse states in the union. Wyoming is the least populous state in the union. And so you can see the primary objectives listed here. The fundamental underlying goal of this entire initiative was to support more equitable access to clinical trials, both to achieve equitable access, but also to ensure that participant samples are as scientifically accurate as possible as well. That was the driving motivation underlying BioNJ's broader health equity in clinical trials initiative. And the lessons learned from this project, both the literal demographic data analyses, but also what we learned from even attempting to do this, have yielded information regarding where there are opportunities for improvement in reaching underrepresented communities, but also where there are opportunities to consider policy measures that could also help to facilitate more equitable access to clinical trials, whether it's improving data interoperability, which you'll hear a lot about, or ensuring broader awareness of resources like those provided by legislation like the Clinical Treatment Act. And we'll get into that towards the end. And so while progress has certainly been made in addressing underrepresentation among demographics, and everyone from companies in the life sciences and the FDA to academic research institutions and the NIH have all acknowledged this as a priority and have made strides, we know that progress remains to be made in achieving accurate demographic representation. And so, next slide, please. We are grateful to have partnered with Medidata, an absolutely optimal partner for a project like this. And I'll introduce our partners with whom we worked in just a minute. But for anybody who's unfamiliar, Medidata is a company that has created software platforms and resources that are used to manage essentially everything related to clinical trials. Their products are used globally. And I think the latest number I saw was that their platforms were used in almost 70% of FDA novel drug approvals last year. And so as a result, the scope and the depth of their ability to scrutinize participant data are both uniquely stellar. And of course, when it comes to, the expertise of the whole Medidata team, they're quite savvy when it comes to data. And so speaking of the Medidata team, next slide, please. We are joined today by Caroline Derr-Ngogoshian, who is a senior clinical engagement lead at Medidata AI Labs with a background in both pharmacy and public health, a PharmD from the Lebanese American University, and a master's in biostats and clinical research methods from Columbia University's Mailman School of Public Health, as well as Yvonne Okidi, who is a senior data scientist at Medidata, who has a background in both biostatistics and data science, a master's in both from Brown University. So in other words, I literally I don't think anybody could possibly have conceived of a better team of collaborators from Medidata. And then before handing it over to Caroline and Yvonne, I would also like to acknowledge the significant contributions of our Grad Futures Fellow, Baris Esser-Yuger. Baris is a PhD candidate in chemical and biological engineering at Princeton University and joined this project right when the rubber was hitting the road. And he truly brought excellent ideas, resources, and just effort to the project, without which I think it's safe to say we likely wouldn't have been able to come to some of the conclusions that we did. So thanks very much for your contributions and joining us on this effort, Baris. And with that, I'd like to hand it over to Caroline and Yvonne at Medidata to dive into the analyses. Caroline.
Yvonne is starting first.
All right. Hi, everyone. I'm Yvonne Okidi. So I'll just go through some of the work that we did here. So we started with a couple of hypotheses. So the first one was clinical trials do not reflect the incidence or prevalence of the disease that they're doing the trial on. So that's, again, based on everything we've seen before, that's the first thing we wanted to do. But then again, because this is like something we're doing in New Jersey, we wanted to see if, despite the similarity in patient population between New Jersey and the other 4 states, New Jersey actually performs differently than the other states when it comes to representing the underlying patient population with a specific disease. So our main objective was to look at the incidence and prevalence data that we have and compare that to the clinical trial composition, demographic composition. And we selected 2 therapeutic areas. This was mainly driven by data availability and how popular those trials were. And so we selected oncology therapeutic area and central nervous system trials in New Jersey compared to all the other 4 states that have similar composition to New Jersey. And because when you talk about therapeutic area like oncology, it covers a lot of oncology trials. And these can vary in how the prevalence or incidence of anything compares between one demographic group to another. So to make it more detailed, like comparing apples to apples here, we went in and selected 4 specific disease areas. So for oncology, we selected breast cancer trials and lung cancer trials. And for central nervous system, we went with Alzheimer's disease and migraine. Next slide, please. And to select a cohort that we were working with, again, here, when we talk of clinical trials, we're talking about clinical trials run on the Medidata platform, so all trial data that we have access to. So when we compiled this for all the 4 disease areas, we compiled this for the 4 disease areas that we're looking for, specific to the 5 states. We also limited this to trials that started after 2010. Again, phase 1, phase 2, and phase 3 trials. Only interventional trials, so no observational trials were included in this, and also closed studies, because open studies have open recruitment, so that demographic composition might change by the end of the study. So we wanted to make sure it's only for closed studies. And this was up to, I think, 2020. And then for the exploratory analysis, for the analysis, we did some exploratory analysis, which is just looking at the data coverage that we have for the therapeutic areas and also for the specific indications. And this was just mainly looking at the clinical trial demographic proportion. So for every therapeutic area, how are each of the specific demographic groups represented? So the proportions of that. So this is very row count. It just tells you what a trial looks like in a state. It's not doing any comparison. For the comparisons, we wanted to, if you remember our objective, we wanted to see how does the trial distribution compare to what the disease distribution looks like. So for this, we wanted to have a metric that could be compared across different states, because different states will have different number of patients recruited in a trial. So for instance, New York might have thousands of patients in a trial, and Connecticut might have just 500. So to make those values comparable, we used a ratio where we're looking at what is the trial proportion and what is the epi proportion. And then finally, just to check how those relative ratios compared across the states, were they statistically different, or could we say they were similar across the states? So we use the chi-square test of homogeneity just to see if New Jersey was same or different from the other states. And if we go to the next slide, I'll go more into detail about what the relative ratio is. I know I was just explaining that it can be used as something that's similar across states. But the relative ratio here was we're looking at the proportion of trial participants for a specific disease. So say we're looking at breast cancer, we look at what is the proportion of, say, Asian patients who were recruited into a breast cancer trial, divided by the proportion of Asians who have the disease in the area that you're conducting that trial. And here, if you had a relative ratio greater than 1, it means that in that particular area for that trial, the clinical trial is recruiting higher than what the underlying disease distribution is. So it's doing way better than what's expected. If it's less than 1, it means that the trial is under recruiting. So it's recruiting less than what you should be expecting from that demographic group. And then because these are estimates, there are a lot of uncertainty around it. So we also added the 95% confidence intervals. And here, I think that this helps us with is to, you'll notice that some confidence intervals are wider than others. So if it's really wide, this was mainly driven by data availability. So the more specific the trial is, you end up having fewer and fewer people in certain demographic groups actually represented. And so that causes us to have a lot of uncertainty around that confidence interval. So I just wanted to show that. Again, because we're using external sources like to give us epidemiology data, we had to use several data sources. One of them was the National Cancer Institute, which was, I think, the most comprehensive source of incidence estimates for this. We used the state-specific incidence information, but they can go as low as like to smaller geographic areas. But in this case, we just use the state level. For Alzheimer's disease, we use the CHAP data. Again, this has been used for a lot of studies, but because it's data that was collected, I think, 2 decades ago, a lot of it is just extrapolation, but to this point, it was the most comprehensive data source that we could find. For migraine data, we used results from the National Health Interview Survey. If we move to the next slide, I'm just going to talk briefly about we're going to talk about this a lot in the next slides, but it was one of the main things that we had to do to be able to use this data. So for Medidata trials, we use CDISC standards for collecting race and ethnicity categories. So for race, we have 8 categories and they're very specific. But then we also, after collecting the race, we also collect ethnicity information. So for every patient, we have race and ethnicity information. For NCI data, the estimates are shown for race and ethnicity categories together. So it will not just be American Indian, it will be American Indian non-Hispanic, Asian or Pacific Islander non-Hispanic, etc. So for us to be able to make the groups comparable, we had to recode our categories from race and ethnicity to map them to the NCI categories. So for instance, if a patient had recorded race as American Indian and ethnicity as non-Hispanic, then they'll be categorized as American Indian non-Hispanic. If their race was American Indian, but ethnicity was Hispanic, they will be recategorized as Hispanic. The same goes for all the other race categories. And as you can see, we have a highlighted section for the group other. So this was something that gave us a lot of headaches just because a lot of other categories ended up being lumped up into this because we did not have complete information. So if your race is American Indian, but your ethnicity is other or unknown, we don't know whether we should classify you as American Indian non-Hispanic or as Hispanic because we don't have that ethnicity information. And so you end up being other. If we have race as other, ethnicity as unknown, again, you just lumped up as other, etc. And if you have race as unknown, but ethnicity is non-Hispanic, this is one of the more common one. We really cannot classify you as any of the other race categories non-Hispanic. So you end up just being other. The only exception was if race was other or if race was like unknown or other, but ethnicity was Hispanic, then you automatically put as Hispanic. So that's like a huge disclaimer that we have to mention here. All right. Next slide, please. So here we did some, remember I mentioned that initially you wanted to look at data coverage and basically the distribution for each race category for the different therapeutic areas and indications. And here we're just showing for one therapeutic area, which is oncology. And you can see that for the 5 states, Maryland is really standing out there as having the highest representation of Black patients in clinical trials. And then followed by New York and New Jersey, respectively. And we can see on the far end that Massachusetts has like the highest representation of white patients in clinical trials. Again, here we did not represent other smaller categories like American Indians, Hawaiian, or a multiple race just because they had less than 1% representation. If we go to the next slide, we're looking at the ethnicity, and this is Hispanic or non-Hispanic patients for oncology trials. And here we see that New Jersey had, just by raw estimate, it had the highest representation of Hispanic patients in clinical trials. And while here we're just showing for oncology, this trend kept up for all the 4 disease areas that we looked at. So the breast cancer, lung cancer, Alzheimer's disease, and migraine. It was always New Jersey had the highest representation and then followed closely by New York. So they seem to have similar representation. And it's important to show this because we're going to go into actually seeing how does that translate when you add the underlying disease distribution. And I'm going to hand it over to Caroline so she can go over that.
Hi, everyone. I'm Caroline, so I'll go over the results section and more of the analysis that we've done so we can move to the next slide. And we'll start with lung cancer. So there's a lot of data here represented on this table, but just to show important things here. If you look at the top, you'll see CT, which represents clinical trial. EPI represents the sample size for the number of patients that were included in the epidemiology data. And here you can actually see that for certain states like Maryland, the cohort of patients that are contributing to the clinical trial data is smaller, and that would translate into some confidence intervals being wider in the next slide. But when looking at the data here as a whole, when we look specifically at Black patient participation, we'll see that all states except New York underrepresent Black lung cancer patients. When we look at Hispanic participation, all states underrepresent this patient population except New Jersey. And if we move to the next slide and look at the forest plot, we can visualize this data a bit better. At first, we notice that there's some wide confidence interval happening for Maryland specifically and Hispanic patients in purple. And that's mainly because we have a smaller cohort of patients contributing to this subpopulation. Therefore, we can take some of these findings with a grain of salt, especially when these confidence intervals are like wider. However, we can see that overall the Hispanic population has also overrepresentation the Asian population has overrepresentation in Massachusetts. But overall, main finding here is that across the board, we see underrepresentation of Hispanic and Black patients across most of the states. If we move to the next slide and look at the breast cancer analysis, here again, a lot of data here that we're looking at, but we had an issue here with the breast cancer analysis, which was described by Yvonne when we were reclassifying from the CDISC race and ethnicity categories into the National Cancer Institute combined race and ethnicity category. And that actually led to an overrepresentation of another category for breast cancer. So when we look here in New Jersey, 45% of clinical trial participants, so almost 50%, ended up in the other category. And then we see that this is a bit lower in the other states, but still a big proportion. This actually, if we move to the next slide, what it led to is underrepresentation of all races and ethnicities across the board in clinical trials. So from a breast cancer standpoint, we would take some of these results with a grain of salt because we can't extrapolate these because of the skewness of the data because of the other category. Moving on to the next slide, here we looked at Alzheimer's disease. So for Alzheimer's disease, the CHOP data that we used only had epidemiology incidence data for Black, non-Hispanic, white, non-Hispanic, and Hispanic. So these three categories. And that's why our analysis was restricted to these three race and ethnicity categories. But overall, from an Alzheimer's disease standpoint, it was very much in line with what is reported in the literature. So in the literature, there's a lot of literature out there showing that Black non-Hispanic patients have a higher incidence and prevalence of Alzheimer's disease than their white counterparts, but they're still underrepresented in clinical trials. And that's actually what we saw here. If you look at the participation in clinical trials of Black non-Hispanic patients, you see a much lower representation compared to epidemiology data. So in New Jersey, we see 4% Black non-Hispanic patients compared to 22% from an epi standpoint. In New York, very similar, 3% compared to 25% from an epi standpoint, et cetera. So again, here we really see that there is underrepresentation of Black patients in Alzheimer's disease trials. And then similarly, we actually saw the same trend for Hispanic patients. If we look at the forest plot on the next slide, it's pretty clear here. We see white patients being overrepresented. So if we look at this data, a relative ratio, so we see this dotted line in the middle, a relative ratio above 1 would show overrepresentation. So clinical trials having a higher representation than epidemiology data, and a relative ratio below 1 showing underrepresentation. And again, here what we see is that almost consistently, Hispanics and Blacks end up being in the underrepresentation category on the left-hand side with a relative ratio of less than 1 and really much closer to 0, right? Like the relative ratio is like closer to 0.25 or between 0.1 and 0.2 for the majority of these. So really showing that there's a lot of room to go from an Alzheimer's disease standpoint to really represent these patients in clinical trials. Moving on to migraine disease. Here, what we actually were surprised with is it was the other way around. So if we look at Black patients in migraine disease, what we actually noticed is that there was overrepresentation of Black patients in clinical trials. So if we look at New Jersey, there was like 31% of the patients in New Jersey sites were Black compared to 14% from an epidemiology standpoint. In New York, it was similar to EPI, but Massachusetts, for example, had 14% of their patients in these sites being Black compared to 5.3% from an EPI standpoint. And then Maryland was similar to EPI. And if we move to the forest plot to visualize the data, again, here what we can see is that whites are maybe overrepresented, but what we still see is that 3 of the states, so Maryland, Massachusetts, and New Jersey, are actually overrepresenting Black patients in migraine trials. And there are some other indications, especially from a psychiatry standpoint, where we have actually seen this as well, such as schizophrenia, where we actually see that in clinical trials, there is overrepresentation of Black patients in schizophrenia trials. So we can move on to like overall conclusion and in the summary of our analysis. Overall, our findings are consistent with the literature. Black and Hispanic patients are underrepresented in cancer, whether it's breast cancer, lung cancer, as well as Alzheimer's disease. In contrast, Black patients were overrepresented in migraine trials, even though in the exploratory analysis that Yvonne went over, it showed that New Jersey had the highest proportion of Hispanic patient enrollment. When comparing that to what we would expect from an epidemiology standpoint, it did not actually overrepresent Hispanic patients compared to EPI data. And that was also held true if we looked at the Maryland data, where Maryland, based on the exploratory data, it looked like it had the highest representation of Black patients. But then when we actually compared to the epidemiology data of these diseases in Maryland, that didn't hold true. Moving on to the data challenges, some challenges that we had here is, as Yvonne mentioned, there is no central database for incidence and prevalence of most diseases. So we used 3 different sources. We used the National Cancer Institute for the cancer data for breast and lung. We used 2 other sources for Alzheimer's disease and migraine. So what that leads to is you don't necessarily have the same categories leveraged for race and ethnicity across different sources. Another limitation is that we had, there is a limitation from a date standpoint. So some of these resources were outdated or are up to 2020. So we were extrapolating these results as they were the incidence and prevalence of today. And from an analysis standpoint, the main limitation was actually leveraging different types of classifications of race and ethnicity. And since all the sources did not have the same standards, we had to do that reclassification, which ended up in a high unknown category. And that leads to limitations in the generalizability of some of these results. Yvonne, Ian, I don't know if there's anything else that you want to add here.
I'm just going to say for a lot of the data, it was out of time, like we said, and that meant we had to extrapolate for that data. And some of it was like the Chicago one is very specific to one region, but then you have to extrapolate it nationally and then make it specific to your specific area that you're looking at. So we also at some point had to bring in census information to help us get incidence estimates. So it was a lot of calculations to get from one estimate and make it useful for your specific disease population in the geographical region that you're looking at. So yeah, that was a major limitation just because you're trying to understand like underlying distribution, but you have to jump through hoops to even get information that's closely even useful. Yeah.
Mhm. And I think, Ian, I'll hand it over to you to go over the policy recommendations.
Sure. Well, thanks again for your partnership on this, Caroline and Yvonne. And thank you again, Barrister Yuger, for your willingness to jump right in. As Debbie likes to say about projects in the past, we were definitely building the plane while flying it. And I think all of the obstacles that Caroline and Yvonne described, while frustrating, were also very instructive. And speaking of that, let's dive into the policy ideas. So next slide, please. Thank you. And just as a reminder, we'd love to hear any questions or comments that you might have via the Q&A button. It's great to see some are already submitted, and I know that some experts in the conduct of clinical trials are here today. So we'd love to hear any thoughts you might have. But as our friends at Medidata described, the distinction between demographic categories was really the primary obstacle that we encountered, beyond, of course, just there's a huge opportunity for more very broad epidemiological analyses to be made publicly available. And we knew that we need to expect this going, that it was going to be a challenge based on prior efforts to improve sensitivity to, public health concerns throughout New Jersey, by expanding demographic sensitivity during the COVID-19 pandemic. And again, as the Medidata team conveyed, there is no central database for incidence and prevalence across the board. And I'm not going to belabor this point further because I think it's pretty clear, but harmonizing these categories would make these kinds of comparisons and this kind of scrutiny of actual recruitment performance and access would be a game changer in our ability to know with sufficient precision where the opportunities for improvement really are, like specifically which communities, literally geographically, are underrepresented for each and every specific trial, and then therefore where to deploy resources to ensure that they have the choice to participate. And then, given the guidance that the FDA has released previously to improve enrollment of underrepresented communities, and the role that the FDA plays in clinical trials, it may very well be in a position to determine these standards, and likely an interagency effort across the U.S. Department of Health and Human Services, to involve the NIH, SAMHSA, CDC, and so on. And so we all certainly appreciate how mammoth of an effort this would be. There are some international standards that might serve as an opportunity to expand that harmonization, that category harmonization beyond US borders, like the Health Level 7 or HL7. This is a set of global standards for the transfer of health data. It's just one example, but that would really be a game changer for this kind of participant, scrutiny of demographics across participants. And so moving beyond the data standards. So one of the many entities with whom we connected in the early stages of this project, and then again, as the project matured a bit, was New Jersey's Medicaid program, NJ, and NJ Family Care. And so, initially we had hoped that the program might have some of their own demographic data to characterize the participation in clinical trials by Medicaid enrollees. And it appears that those data do not exist. And frankly, it's unsurprising because unless the state's Division of Medical Assistance and Health Services, which administers the Medicaid program, is both given direction to collect those data, but then also given the resources that would be required for them to do so, it's unsurprising that this kind of data collection would not be performed, or at least not compiled in a way that would make this kind of scrutiny performed in this project possible. And so, ensuring that the state's Medicaid program has the resources to do so would help to ensure that everybody has a sense of whether their, enrollees are underrepresented in clinical trials. But another issue, policy, topic associated with the Medicaid program is that, from what I can tell, there doesn't appear to be a broad awareness of the implementation of the Clinical Treatment Act. And so this was a bill that was signed into federal law in December of 2020, and it took effect at the start of 2022. And what it does is it requires all states and territories in the United States to cover and reimburse routine costs of care for treating a Medicaid enrollee participating in a clinical trial. So that includes items or services that are provided to prevent, diagnose, monitor, or treat complications resulting from a participation in a trial. And that includes the cost of, physician visits and, diagnostic lab tests, right? And so, it may very well be the case that I wasn't able to connect with the folks who are aware of this legislation at the department or the division, and I'm quite confident that there are some who are aware of it. I would be shocked if there weren't. But just from my own interactions with the kind of, frontline people who literally, pick up the phone when an enrollee might call, it certainly seemed like even the idea of an enrollee participating in a clinical trial was just not on their radars. And so, empowering the state's Medicaid program to be sensitive to understanding how their enrollees are accessing clinical trials, but then also ensuring that everyone involved in the Medicaid program, whether, either within the department or as an enrollee, that they're all aware that they have access to the latest innovations in biomedical science. Both of those would very likely help to ensure that communities who remain underrepresented in trials at least have the choice of whether they'd like to participate, when eligible. And then moving on. So the next is to at least consider the roles that other demographic characteristics might play in both health disparities as well as potentially disparate trial participant outcomes. And in particular, sexual orientation and gender identity. And so in the white paper, we include some specific examples, some citations from the academic literature of how these categories have been associated with disparate health outcomes. And while we know that there have been some efforts to explore these categories in greater depth than they typically are, and we know that even harmonizing data categories alone, will be a very heavy lift. Just stipulating both of those realities, this is certainly a dimension of health and demographic variability that can play a role in health and trial outcomes that can really matter. And so moving forward, it became clear that certain recruitment sites likely account for a disproportionate share of demographic representation across trials, right? Whether because a uniquely high number of a specific community have been recruited, or because a particularly low level of communities are being recruited, and that's unsurprising, right? But understanding recruitment performance at specific sites, whether that performance is put into the context of demographic characteristics of the communities in proximity to those sites, can really help to hone in on where the most efficient use of outreach and engagement and resources can be applied. And then beyond that, understanding if a particular site or institution is very effectively recruiting any specific community can, of course, yield insights, regarding what it is that they're doing to achieve those outcomes, what they're potentially doing right, as well as opportunities to deploy more effective best practices. Right. And so, again, of course, the white paper elaborates quite a bit on essentially everything we've discussed today. And of course, there are always additional data sets that various institutions and entities have at their own disposals. Right. But I think I speak for the team in saying that, we're proud that we've done this rather granular assessment of demographic representation. And I should note, we started literally at the zip code level across the state. To see if there are literally regional disparities or disparities that occurred at a regional level. We learned quite a bit through this process, and we hope that this ultimately provides at least some food for thought just for the recruitment efforts themselves at a minimum, the recruitment performances or teams, but also sheds at least a bit of light on how the obstacles that prevented an even broader and more precise assessment could have been made, and ideas for public policy to diminish those obstacles, right? Because, ultimately what we're talking about is ensuring every single patient, no matter who they are, where they live, is able to have the choice of participating in the latest biomedical innovations that they know they can, and they know when they can. So I'd love to open the floor up to any questions or comments that anyone might have via the Q&A feature down below. And again, I know there are some folks who are extremely experienced in clinical trials who are in attendance. So any insights, any thoughts you might have would be very appreciated. But starting with the first questions that were submitted, I'm sorry, I have to scroll up. So how does this program, or how can this program, work with sovereign nations such as Cherokee tribes or nation to conduct clinical research within the community, out of their facilities in New Jersey, et cetera? So, I mean, it's a great question. And I think the granularity of conducting an analysis like this can help to identify where communities like the Cherokee tribes or nation and similar communities across the country, frankly, but certainly, in our region, are underrepresented, right? But in an ideal world, could also help to identify which recruitment sites are best situated to connect with those communities, to ensure that, every single member of those communities has the choice and knows that these opportunities to participate in these trials are available. But Caroline, Yvonne, I don't know, do you have any other thoughts in reaction to that question? Okay, so moving to the second. So the second is, how can we incorporate Schedule I drugs such as cannabis, psychedelics, ibogaine, to conduct research, as many of these compounds have less side effects, if any, and have profound effect on addiction, PTSD, etc. So, I'll be honest, my reaction is the kind of work that we described today isn't necessarily focused on determining optimal potential therapeutic candidates. But once again, I think this kind of an assessment of participant demographics would help to reveal if and whether, there might be communities who have been uninvolved, underinvolved in, assessments of the effects of compounds or the efficacy of compounds that you're describing. So, just like with any therapeutic question, if we want to develop an accurate sense of the effects of these compounds, we want to be absolutely certain that we're basing that sense on, as accurate a sample as possible, right? And as the white paper we, released earlier, which is publicly available, you can all check it out. Again, it has greater detail on all these points. As the paper, explores, it is very well established that different communities respond differently to various compounds. Right. And so that's my reaction. I don't know. Again, Yvonne, Caroline, I don't know if you have any thoughts in response to that one.
But I agree with what you said. I think it's just any evaluation would also need to include a representative patient cohort, and thinking about diversity and incidence and prevalence of, let's say, PTSD or any of these diseases, to identify where these patients would be, and evaluating these agents in a representative cohort of patients would allow results that are generalizable to the population as a whole.
Excellent. Well, thank you, Caroline. Okay, so moving to the next comment, I believe. Yes. Great presentation. Thank you very much. Have you analyzed the distribution of participants across age cohorts, further broken down by race and ethnicity? That's a terrific question. I'm going to turn that right over to the metadata team.
Yeah.
Yeah, so we do have in our clinical trial data, we have information on age, race, ethnicity, et cetera. Typically, I think we've looked at age, but I don't think we've done age, and at least for this, we haven't done age broken down by race and ethnicity. And one of the things that initially jumps out at me is just when you slice the data into such small categories, you end up having issues of not enough representation. I mean, even right now, when we're just looking at specific CNS categories, you could see in our tables that we had American Indian not represented in both clinical trial and epi incidence rate. So it becomes very little. So if you take that again and bucket them into age categories, it gets very small. So this would be good if you're doing bigger view, like if you're looking at maybe the US or bigger categories, so we have more data rather than just specific states. But yeah, it is good, and we'd also need an indication that has a wider inclusion exclusion criteria. Most of these indications tend to recruit, like Alzheimer's, you're getting older people. Breast cancer, it's mostly restricted to like female, like if you're looking at sex, it's restricted to female for most of the trials. So again, it's good, but we have to be very specific on what we're looking at and mindful of data limitations, unfortunately.
Awesome. Thank you both very much for responding. As you can tell, this is something we absolutely contemplated and explored whether it be viable. But it's a great point, and hopefully we can make progress on being able to scrutinize data at that level sooner rather than later. Next question, and I think this is going to go right back to you both. Can AI or machine learning analysis of electronic health data help to solve some of these issues or problems? I happen to know that Medidata is interested in artificial intelligence. So do you have any reactions?
Yeah, that's a good question. I think from a categorization standpoint, electronic health data, from a classification standpoint to CDISC, we're good from that aspect. I think it's the fact that open sources for epi data don't represent all these subcategories with the same granularity. And that's actually the main limitation that we faced, where we were actually not able to have the same level of granularity in epidemiology data. So we had to reclassify. And by reclassifying, we ended up having a large pool of patients who fell under another category for a race and ethnicity. But Yvonne, back at you. I'm sure, yes, we can use AI and machine learning for classification.
Yeah, for sure. Like it with enough data, it for sure helps. And this is something we've been thinking about just because a lot more people are interested in having diversity information. Like if you're recruiting, you want a representative sample, etc. The thing about electronic health records and like real world evidence, I think we've done a similar thing with Caroline on using real world evidence to see how if you can actually recruit patients where they are seen by their primary care physicians or whatever. It does not always translate. And again, it comes back to the issue of collecting that information. We end up with a lot of unknown and like missing or other for real world evidence because it's not always required. For clinical trials, we tend to have it as a requirement sometimes in certain forms, depending on which part of the world you're in. Some EU countries don't really collect that. But yeah, it might be easier to do it with clinical trial data just because it's more complete and populated. But yes, AI and machine learning methods are absolutely probably the next step to trying to fill that gap.
Well, thank you both. Moving to the next question. How is the new Trump incoming administration policies, et cetera, affect all of this overall? It's a great question. Obviously, I will take this unless you both have any thoughts to contribute. But we don't know a lot about what leadership will look like. Leadership of, for example, Health and Human Services, under which FDA, NIH, essentially everything that we discussed today falls. But I will just give you this one data point. I mentioned a bill called the Clinical Treatment Act, right, which essentially requires Medicaid programs to cover trial participation among their enrollees. That was signed by former President Trump. And so, I think it's tough to know until we see, what leadership will begin to look like. I think we'll start to see that in, the coming weeks. But without a doubt, whomever is in the White House, whomever is, running HHS, FDA, plays a crucial role in determining all of this and well beyond, this conversation. Okay, next question. So the comparison between prevalence, incidence, and trial participation is really insightful. Did your research look at specific barriers to access within each racial and ethnic group? For example, are there differences in awareness, physician referrals, or trial locations that might be contributing factors? Also, are there other successful outreach or recruitment models from other states or regions that could be adapted for the New Jersey context, including policies? I mean, that's the next white paper, like that 100%. These are conversations that we had. Our white paper, our mission did not include exploring the barriers, right? It was more identifying where those barriers are. And, that said, another, you mentioned, physician referrals, all these kinds of things. Another that we contemplated and almost included in this was, the demographics of the PIs running these trials. We know that that plays a huge role in just, recruitment performance. And so, the broader initiative, BioNJ's broader health equity and clinical trials initiative did explore that. And there are some best practices that are available on BioNJ's website, contributed by many of the companies who sponsor these trials. But it is, it's a great point, and I think this is one of the several of the first steps in identifying, okay, wait, this region or this site is performing uniquely well in recruiting, any given community. What can we learn from them? I think that is what this project, this component of the initiative, was intended to fuel, essentially. But I don't know, do either of you, Caroline or Yvonne, have any thoughts?
Yeah, I think the main interesting finding that we've had with this analysis and other analyses is that actually having a site in a geographical area where these sub-patient populations are located doesn't necessarily improve the chances of enrolling these specific patient populations. So there are still barriers, despite the site being in a good geographical location to, let's say, recruit Hispanic patients or recruit Black patients or Asian patients. This does not necessarily translate into these clinical trial sites enrolling these patients. So thinking more about these barriers that these sites have, whether it's translating their consent forms to Spanish, if you're trying to enroll these Hispanic patients, or having PIs that are representing or of a demographic subgroup that represents these patients that you want to enroll, may be more helpful. But definitely there's a lot more work to do to really understand more the reasons or contributing factors for these differences, especially when we see that migraine has actually the other way around, right? Like overrepresentation of Black patients. So really learning the differences in these recruitments across different indications and learning from them.
Excellent. And then the second part of that, are there other successful outreach or recruitment models from other states or regions, right? What I can tell you is this initiative, this project started with very broad outreach to as many institutions as we could possibly think of. And I think we covered a lot of ground there. And I do know that there are some institutions within New Jersey who are really pretty close to hitting their marks. And so, yeah, what we would hope is that the part of the motivation of this whole initiative was to bring the whole community together and evaluate best practices, see what's working, see what's not working, or just identify opportunities for improvement. So yes, the answer is yes, there are, even within New Jersey, but these are not necessarily the same communities that interact with each other all the time. So yeah, hopefully, the material in this work, in this analysis, can fuel that kind of outreach. Okay, where is the performance? From whom can we learn best practices? Okay, next question. So do we know if there are recruitment sites in a local geographic area to the population that is underrepresented? Are we seeing overrepresentation of patients enrolled in areas that have large academic centers that may have an increased attraction to a wide diversity of patient population? I think I know what Caroline or Yvonne might say, but please, I'll let you jump in first.
Yeah, not part of this analysis, but part of another analysis, we were actually looking at performance of site type, because the hypothesis was that actually the other way around, that community sites are better at recruiting certain demographic subgroups, and academic medical centers not really. But actually that analysis didn't hold true. We were not able to find that a certain type of site, it has a better representation of demographic subgroups. I don't think that we've really pinpointed reasons for a site or other factors that are actually leading a site to perform better than another one from a diversity standpoint. But I definitely think that there's probably so many different reasons that have already been shared in the literature, whether it's like mistrust from patients or not having PIs that are representative of the patients that we're trying to enroll, language barriers, etc. But we haven't looked into these specific factors. But from a site type perspective, we had a hypothesis, but we actually were not able to find that a specific site type is better at enrolling diverse patients.
Yvonne, or did you have any other thoughts? No, yeah, okay. Yeah, that's, I'm unsurprised by your reaction. And this does kind of speak to the final question, which is, it is mentioned that the migraine clinical trials, Black patients were overrepresented. What were the key reasons for this compared to the other diseases that you researched? I think, Caroline, you just spoke to this, where we don't necessarily know, but you know, I don't know if you have any other thoughts beyond that.
We worked with another collaborator on a schizophrenia analysis, and in schizophrenia, patients, like Black patients, are overrepresented. And one thing that his hypothesis was that certain indications have these consortiums of sites and PIs who have cracked the code, and they really know how to get to these demographic subgroups, and they enroll these patients much easier, any patient population. So let's say, like, for schizophrenia, we're speaking of Black patients, they have cracked the code at like getting to these patients and enrolling them. But I think we definitely need to analyze that further to figure out if like that hypothesis holds true.
Awesome. And I am just going to rush through the last comment. Thank you so much for it. What policy coming out of this important project to allocate funds to do more studies on patient barriers can be proposed to Trenton on the state level? Could the funding be around AI or machine learning? Well, so at the end of the white paper, we list all of these policy proposals, all of them. Now, you know, the data interoperability issue, that is almost certainly more of a federal challenge, because it would require that level of just coordination. But all of them, I think, are viable, and we're going to be working on all of these policy issues, bringing them up with our policymakers in Trenton. But we are almost quite literally out of time. So thanks again, everyone, for your attention and care for this issue of equity. And once again, I just love to thank, you know, Medidata once again for your partnership on this. And thank you again, Barris, for really hitting the ground running and joining this project when you did. It was quite a journey. And it's always lovely when someone can learn from their colleagues. And I certainly learned from all of you. But with that, I will hand it back to Debbie to close the program.
Thank you, Ian. And I am learning from you on a daily basis. Thank you, Ian, Caroline, Yvonne. Thank you, Barris. Thank you to Medidata, and just an amazing, extraordinary partner in this process. You know, obviously lots more work to be done. I'm sure there are lots more questions out there that have not been asked, but need to be asked. And so to our knowledge, this report is the first of its kind, but hopefully not the last, since again, this is an important topic. And as we just heard, there's so much more to be done that, you know, to bring health equity in clinical trials. Our collaboration with Medidata on this white paper aims to strengthen recruitment, ensuring all demographics have access to biomedical advances, and that new therapies are effectively evaluated within the communities they serve. And we, again, could not have done this without the expertise and considerable commitment of Medidata, specifically, again, Caroline, Yvonne, Barris, and Ian. So thank you and congratulations for the work that you've produced. More to come, I'm sure. And for the full report, you can take a look at bionj.org. And again, the recording from this session today. If you're not yet a member, I have to say this, not yet a member of BioNJ, please contact Sherry or Danielle. We would welcome having a conversation with you. It enables the work that we do. Before you sign off, if you wouldn't mind taking just a moment to scan the QR code on the screen to complete a very brief program evaluation. Your feedback is just so important to us, and we very much appreciate that, as well as your participation today. I hope you have a wonderful rest of the day.